Good morning, good afternoon, and good evening, and thank you for joining us for the second event of our Animal-Free Workshop series for early career researchers. I'm Michaela Singer from the Physicians Committee for Responsible Medicine, a nonprofit advocacy organization based in Washington, D.C., focused on preventative medicine and improving human health through research, testing, and education without animal experimentation. This workshop series is part of our ERA 21 program, which is designed to educate and support researchers committed to advancing science using non-animal methods. Beyond this workshop series, we offer travel awards to help scientists presenting non-animal research at conferences and host the Summer Immersion on Innovative Approaches in Science, which is a free, in-person, immersive 3-day event offered every other year. The next Summer Immersion will be in June 2026. Be sure to sign up for our newsletter to stay up to date on these events. I'm going to pass it over to Kat Herman of the Center for Alternatives to Animal Testing for a brief introduction on the center now. Over to you, Kat.
Hi, thanks, Michaela. Hi, everybody. Nice to see some of you again. I think some of you already attended the first workshop we hosted back in November. I work at the Center for Alternatives to Animal Testing at Johns Hopkins University, and there we promote humane science by advancing animal-free methods in research, safety testing, and education. And CAAT is actually one of the oldest 3R centers that was already founded in 1981 at the Bloomberg School of Public Health, and it's actually an interesting story to it because the money for the establishment of CAAT came from animal advocacy against animal testing by the animal advocate Henry Spira, who managed that the Cosmetics, Toiletry, and Fragrance Association gave $1 million at the time to establish CAAT. And I will just post our newsletter link in the chat shortly for you, for those of you who don't know the center yet. We also have some funding opportunities, and we have free webinars and workshops like this one. And yeah, we want to do more to to accelerate the shift towards humane and human relevant methods. Thanks for being here, and I hand it back to Michaela.
Thanks, Kat, and thank you again, everyone, for being here. During today's workshop, if you have any questions, please use the Q&A feature to ask them. Now, I'm pleased to introduce our speaker today, Dr. Esther Wenzel. She is an expert in animal-free technologies. She is the co-founder and COO of the biotech company Abcalis that specializes in animal-free antibody discovery and production and offers consulting to find solutions for animal origin work in labs. Since the early 2000s, Dr. Wenzel has gained extensive experience with animal-free technologies of 3D cell culture and spheroids, has perfected high-throughput methods for the functional testing of antibodies, and co-invented multiclonal antibodies in animal-free replacement for polyclonal antibodies. Welcome, Dr. Wenzel.
Thank you very much for this kind introduction, Mikhail. So I'll share my screen. And please give me short feedback if that's working.
Yes.
Perfect, thank you. Good morning and good afternoon, everyone. My name is Esther Wenzel, and I'm co-founder and Chief Operating Officer at Abcalis. In this position, I'm responsible to make sure that the day-to-day business runs smoothly in our business. So I'm a biotechnologist, and we are a biotech company developing animal-free antibodies for diagnostic and research. We are based in Braunschweig, Germany. That's between Hanover and Berlin. And it's also the region where the headquarters of Volkswagen or Jägermeister is based. Today, I'm excited to talk about the power of animal-free recombinant antibodies. I hope you'll find it interesting and useful, so let's start. My vision and also the vision of my company Abcalis is a world without the use of laboratory animals. So we like to remove every hidden animal-derived reagent from our lab to have a really animal-free lab. So how we do this, I will talk later. But let's start with the main topic of this workshop, antibodies. Antibodies are the key component of most of our experiments in the lab. So if you do an ELISA or tissue staining, immunohistochemistry or Western blot, or we all know it from Corona, a point of care test like a Corona test. In all of these assays, the key component is an antibody. But before we start to dive deeper into the topic of antibodies, I would like to know a bit more about you, my audience today. So therefore, our today's poll platform is WooClap. It's like Slido, most of you should know that. So what I would kindly ask you to do is take your smartphones or tablets and scan the QR code. Then you should immediately come to the next poll. And if you don't have a smartphone or a tablet, you can also join via browser. You just have to type wooclap.com into the browser and then enter this 3-letter event code on the top in the banner. Then you would also see the next poll. I give you just some minutes, some seconds to join. I already see there are 62, 63 people in. Increasing number, perfect. So seems to work pretty well. Yeah, perfect. So wait another seconds and then we'll start with the first question. So, and you should now see the question on your screen. So my first question is, where are you from? So where are you locating at the moment? Oh, pretty nice. Yeah, I'll give you another 5 seconds. Perfect. I think most of you voted. So I close that question. And the next one is, what career stage are you in at the moment? What describes your career stage best? Perfect. I'll give you 5 another seconds. Okay. Come to the next question. And this is, what's your field of study? So mine was biotechnology, for example. I'll just do this way. Oh, really nice. So we have a really diverse field, a really interdisciplinary field like that. So I'll give you another 5 seconds for that answer, for answering. Okay, then let's move to the next question. So if you're using antibodies, if you're working in the lab, for what application do you use antibodies normally? And multiple answers are possible in that question. I would give you another 10 seconds just to see if more answers are coming in. But we already see a lot of you are doing immunochemistry, ELISA or Western blot, so the typical applications, and flow cytometry for using antibodies. Huh? Perfect. So let's come to the next question. And this is, how would you rate your knowledge of antibodies or about antibodies? So I would wait another 10 to 15 seconds. Nice to see that most of you have already a basic understanding of antibodies. Let's see if we can increase that today in the workshop. So I will close that question. And let's come to the next question. For me, the more important one. Have you ever worked with animal-free antibodies? Give you another 10 to 15 seconds. So I already see how you voted. So you see that too. So I hope most of you, so 2/3 of you have not worked with animal-free antibodies beforehand. So let's see if after that workshop you consider animal-free antibodies for your work. Hope so. So many thanks for your active participation in the polls. So now there will be a bit of lecture, but don't put your smartphones too far away. There will be a few questions more later on. So But now let's talk about antibodies. What is an antibody? How is it structured? Why can we use it for our assays? So, good. So, okay. Antibodies or immune globulins are Y-shaped. Their structure is composed of several distinct regions, each with a specific function. This is an immunoglobulin G, short IgG. It's composed of two identical heavy chains, here in gray, and two identical light chains, here both in green. These chains are connected by disulfide bonds, forming the Y shape of the molecule. We have the variable fragment. This region represents the part of the antibody that's responsible for the antigen recognition. It includes the variable domains derived from both heavy and light chains. These domains contain the complementary determined regions, short CDRs, which forms the specific antigen binding site, allowing the antibody to recognize a unique epitope on the antigen. Then we have the Fab fragment, encompasses the variable fragment region, along with the portfolio of the constant domain from light and heavy chain. This region forms the two arms of the antibody's Y shape and is directly responsible for the binding of the antigen. Each Fab fragment is identical, enabling an antibody to bind the same antigen simultaneously with both arms. Then we have the hinge region, dislocated between the Fab and the FC region. The hinge region provides flexibility to our antibody structure. This flexible segment allows the Fab arms to move relative to each other, enabling them to adjust and bind antigens at varying distances and orientations. The hinge mobility is essential for the effective antigen binding and subsequently for the immune response too. And in the end, we have the FC region of the antibody. The FC region is the constant part of the antibody's heavy chains that form the stem of the Y. While it does not participate directly in the antigen binding, this region interacts with the cell surface receptors, the FC receptors, and other components of the immune system. It's mediating effector functions such as antibody-dependent cell-mediated cytotoxicity, short ADCC, and it makes complement activation in the immune system. For most scientists, this FC part of the antibody will be of interest because in lab, we use that to detect our primary antibody. So, central to their ability to recognize a vast array of antigens are these complementary determined regions, short CDRs. These specialized regions found within the variable domains of the antibody molecule. They are key to the specific and diversity of antigen binding. The variable regions of these chains where antigen recognition occurs, so here, contain this hypervariable loops. So the loops you see here, known as CDRs. They're typical 3 CDRs on each chain, so on the heavy and the light chain. Yes, you see that here. At the molecular level, CDR forms the loops that create a unit binding pocket on the antibody surface. The conformation of these loops determines the shape and electrostatic properties of the binding site, which influence the antibody's ability to interact with its corresponding antigen. So let's talk about the structure of the CDRs. Here you see again the CDRs 1 to 3 for each heavy and light chain. The variability of CDRs is generated through a process called VDJ recombination during the B cell development. This mechanism results in a diverse set of gene segment recombining to form the variable regions. Mutation and somatic hypermutation further enhance the variability particular to the CD3 region. So here we have the highest variability of the CDRs, mainly in the CDR3 region, and this allowing the antibody to bind with high specificity to a huge variety of antigens. So the primary function of CDRs is to mediate antigen binding. They do this by directly interacting with epitopes of antigens. So in our immune system, during the immune response, the specific arrangement of amino acids to the CDRs determines which antigen can be bound. The shape and charge distribution of these regions enable them to fit like a lock and key with the antigen's epitope, ensuring specificity. This high degree of specificity is what makes antibodies both effective in neutralizing pathogens in our immune system, but this specificity is also the reason why antibodies play such an important role in our daily laboratory life. By tailoring these CDRs to target specific antigens, scientists can create highly sensitive diagnostic and research tools that are crucial for our detection systems. So, the CDRs are essential components of the antibody structure, providing the necessary diversity and specificity to recognize a large number of antigens. Their unique structure created through highly regulated genetic processes forms the cornerstone of effective immune response and has significant applications in diagnostic and research. So, but how do they interact? So how do the epitope and the paratope interact? This binding is driven by several non-covalent forces that work together to achieve specific and stable interactions. For example, we have hydrogen bridges that are formed between polar groups of the amino acids in the paratope. Paratope is always on the antibody side, and the corresponding group in the epitope on the antigen side. Then we have hydrophobic interactions, so non-polar regions in both the antibody paratope side and antigen epitope side tend to associate in aqueous environment, stabilizing the complex through the exclusion of water molecules. We have diionic interactions, so the attraction between oppositely charged residues on the antibody and antigen side that can enhance binding strength and specificity. Then we have the interaction of the p-electron system, so aromatic amino acids like phenylalanine, tyrosine, or tryptophan in the paratope and epitope side can engage in stacking interactions between their aromatic rings. This interaction helps stabilizing the complex through the overlapping p orbitals. And last but not least, we have van der Waals forces. Although they are individually weak, these interactions collectively contribute significantly to the overall affinity through close contact between the binding surfaces. So combining all of these forces, each contributing its part, ensures that the paratope can recognize and bind specifically to the epitope with a high precision. So I told you about the loops, and this is just an example. So this is a German football goalkeeper, Manuel Neuer, and just think that his fingers are the loops of the antibody, and the ball is the antigen. So this as an example that when you think of that, his fingers, so the loops of the antibody, always can catch the ball to get a high specificity of antibody and antigen. So this high specificity of an antibody arises through the unique three-dimensional structure of this variable regions that forms the paratope. So the loops, the 6 loops, 3 coming from the CDR loops, 3 coming from the heavy chain, 3 coming from the light chain, like I explained it in the last slides. This structure is complementary in shape, charge, hydrophilic, hydrophobic properties to their specific epitope on the antigen site. So on one antigen, various epitopes can exist. So different antibodies, antibody A and antibody B, could bind to one antigen, but on different epitopes. So if a similar but not identical structure occurs in a second antigen, our antibody will also bind to this epitope. This we call cross-reactivity. This happens due to structural or chemical similarities among different epitopes. So, different to that, we also have unspecific binding. This refers to antibodies attaching to targets or surfaces indiscriminately. So rather than binding to its intent epitope, it's not to a specific lock and key fit, but rather arises from a non-specific physical or chemical interaction. This unspecific binding can lead to background signals or false positive in diagnostic and research experiments. So this is why it's so crucial to optimize your assay conditions and use proper controls to minimize these interactions, these unspecific interactions when doing your experiments. So I explained you how an IgG, so immune globulin G, is structured. And if you just see it in single components, you have six isolated structures. You have the variable light chain and variable heavy chain here in red. You have the constant light and heavy chain 1. So these builds up the Fab. And then you have the FC part with the CH2 and the CH3. So these you can combine in really a lot of variations. So there's a combination experiment made by Chris Higgins already in 2017, published. And he asked how many combinations are possible with just 6 bricks. And just when you just think about a number. Just combining each of the so 6 bricks in each in each position you can imagine. And what he figured out mathematically is that over 900 billion possibilities are there. Combine just 6 Lego bricks. And this is why we have this huge variety of IgG antibodies also in our body available. So our body is just combining the ground genetic information and is doing a completely new IgG. With that, each time it's challenged with a new antigen, so pathogen, for example. This is how we build up antibodies in our immune system. So, and we cannot just make IgG antibodies with that. There are also pentamers like IgM antibodies or IgA antibodies that are 2 nearly IgG-like antibodies combined. So just as an example, what other type of antibodies are possible. So this is just how an antibody looks like. How is the structure of an antibody? So now let's talk about antibody generation. And just to give you a short overview of the types of antibodies that are normally commercially available. So on the left side, there are different types of animal-derived antibodies. And on the right side, there are the animal-free antibodies. So to show you the differences between the various antibody types, I will take you through development of these antibody types in the next few slides. And we start with the animal-derived antibodies, the polyclonal antibodies. So to generate a polyclonal antibody, you have to immunize an animal, usually several times, and the animal's immune system will produce antibody against the injected antigen. After 6 to 12 weeks, the blood is drawn from the animal. And in the blood, the serum contains the mixture of the various antibodies produced from the immune system of the animal. For the generation of these polyclonal antibodies, different animals are used. These animals are selected based on their immune system's ability to respond to a specific antigen or to the required quantity of the antibody. If you need a lot of antibody, normally a huger animal is used to get more serum out of that. So in the European Union, we count the numbers for regularly production, so just the production companies dealing with polyclonal antibodies. Here, just in the European Union in the year 2022, nearly 185,000 animals are used for the generation of antibodies. So this is not just the polyclonal antibody generation, this is a combined number. This is a combined number of the also monoclonal antibody generation I will talk in the next slides about. But first, let's stay with the polyclonal antibodies. The disadvantages of these polyclonal antibodies are really obvious. So we have first the specificity issue. So polyclonal antibodies are a mixture of antibodies that bind to various epitopes on the antigen, but also these antibodies are all coming from the immune system of the animal. So there is a huge diversity in the whole antibodies. So this can lead to cross-reactivity, for example. I explained a few slides ago, so that we have a similar antigen because we have a huge variety of epitopes given to the animal. But also this can result to really non-specific binding and high background noises because we have a lot of different antibody in this mixture. It's a really undefined mixture. It's all coming from the serum of the immune system of the animal. So, but also the limited supply is a problem. So polyclonal antibodies are harvested from serum. So once the supply from a particular animal is exhausted, producing a new batch requires new immunization and from the same or a new animal. So one batch just lasts as long as you have serum from one animal. But this also leads to an inconsistency between the batches. So each batch of polyclonal antibody is derived from a different animal or so a different immune system and different immune response, or the same animal but to a later time point, so also different immune response. This is leading to a various so to really undefined mixture and an inconsistency that can also affect your experiments and results if you buy a new batch of antibodies. So next coming to the monoclonal antibody generation using mainly hybridoma technology. Here, an antigen is injected normally into a mouse, and also that several times to stimulate the immune response and therefore the production of antigen-specific B cells. After the mouse has developed a strong immune response, the spleen is removed to isolate the B cells that produce our wanted antibodies. These isolated B cells are fused with myeloma cells, so tumor cells, because they can proliferate infinite. The fusion then results in a hybrid cell called hybridoma cell. These cells combine the antibody producing capability of B cells and with the immortality of myeloma cells. So we have an infinite source of a single B cell that can produce one antibody. This positive hybridomas so now other way around, then we have to be sure that the positive hybridoma produce our desired or wanted antibody, and that this wanted single cell is selected. Here we have to guarantee that it's a single cell because this is the aim of a monoclonal antibody. If we have our monoclonal, or our single cell that produces a monoclonal antibody, this selected monoclonal hybridoma cell can expand it in cell culture to produce higher quantity of this antibody. The antibodies are harvested from cell culture supernatant and mainly purified with affinity chromatography with protein A or protein G. This ensures the production of antibodies that are highly specific to a single epitope and can be produced consistently in large quantities in cell culture. So still in use is also the so-called ascites method to produce monoclonal antibodies. So here we also have the hybridoma cells that also derives from the spleen of a immunized mouse, but they are not produced in cell culture like beforehand I explained. They are injected into the peritoneal cavity of a mouse. Then over the next 1 to 2 weeks, the hybridoma cells proliferate within the peritoneal cave. Yes, the mouse begins to produce ascites fluid. This ascites fluid accumulates in the peritoneal cavity and contains a high concentration of the monoclonal antibody. So it really develops a tumor here in the peritoneal cave. This ascites fluid is harvested by periodically tapping the peritoneal cavity with a needle and a syringe. Depending on the health of the mouse and the volume of the ascites fluid that's produced, multiple collections can perform over a period of several weeks. The supernatant that we harvest contains then the antibody and can also further purified with affinity chromatography with protein A or protein G to isolate the monoclonal antibody from other proteins and contaminants occurring in the ascites fluid. For this method, so I just have the numbers again for the European Union. In 2022, nearly 50,000 mice were used per year in the European Union, still with that, producing any antibodies still with that method. So now I talked a lot about monoclonal antibodies and how important it is that monoclonal antibodies are specific and targeting just one specific antigene, so one specific epitope to be precise. So I would just shortly go over a paper published 2018 by Bradbury et al. You can find the publication here in the QR behind the QR code also. And they made a multicentric study of several random chosen hybridoma sequences. So what they did, they just had a look at the genetic information of the hybridomas that are in catalogs and analyzed if there's really behind that hybridoma clone a monoclonal antibody. And what they figured out was that over or nearly a third of all monoclonal hybridoma antibodies in the catalog are not monospecific. What does it mean? That means that they don't just produce one heavy and one light chain that is combined in the perfect way. So like we see it here, one heavy and one light chain. They produce an additional light chain, for example, that can combine then in a way that we have one arm with a correct and one arm with a false light chain, or even both arms with a false light chain, so that we have another specificity there, because I explained also to you several slides before, we have this CDR region that forms the fingers to catch the antigen. And if you think about that, half of the fingers are not doing the thing they're supposed to do, but doing something else, they can also bind to something else. And then this antibody is not longer monospecific. So, how did they do that? So, what was done by them to find these unwanted byproducts? So, they converted the hybridomas. So, this leads us also to our next antibody format. These are the recombinant antibodies. The normal way at the moment to get recombinant antibodies is the conversion of hybridoma antibodies. So we still have the immunization of the mouse that was done, where we got the hybridoma cell from. And if you have the hybridoma cell, so the easiest way is to extract the mRNA from these hybridoma cells with just standard RNA extraction techniques. With that, so this mRNA then contains the genetic information of the antibody, of the heavy and the light chain, and you can easily produce a recombinant antibody with that. If you just have the produced antibody, also via proteomics and mass spectrometry, the antibody can be sequenced and you can identify an amino acid sequence out of that. And this can also lead to the DNA sequence that provides us the sequence for a recombinant antibody that can afterwards be produced. So just for the sake of completeness, there are also companies out there that say they can do this proteomic thing with polyclonal antibodies. We never tried this, but there are several publications, and I also believe with improving AI techniques, it's becoming more easier to get also from polyclonal antibodies the sequence, the amino acid sequence to make with sequence design, then DNA sequence available and produce also polyclonal mixtures. But the thing is, why should you do that? So, and what we alternatively also can do is we can extract or use the B cells of animals that are already immunized and doing phage display, creating a phage display library with them, doing phage display. So, but this brings us finally to animal-free antibodies. So, to develop animal-free recombinant antibody, you need the genetic antibody information from non-animal derived B cells. This information is used to create so-called display libraries, and they can be used in different display technologies, for example, phage display, yeast display, mammalia display, or ribosomal display. So in my next slides, I will focus on the generation of animal-free antibodies using phage display, because this is the technique we use at Apcalis. And this technique also won the Nobel Prize in 2018 in chemistry for phage display of peptides and antibodies. So, what is special about phage display and getting animal-free antibodies out of that? Why can we use that for developing the animal-free antibodies? So, in phage display, we use this variable fragment. This region is specific for each antibody because it contains the CDR regions that are the responsible regions for the antigen binding. And we turn that into a so-called SCVFC, so a single chain variable fragment. So what we do is we connect the variable fragments for the heavy and the light chain with a linker. So we take this fragment and display it on the surface of a phage. With that step, we link the genetic information, so the so-called genotype, with the protein on the surface, the so-called phenotype. So, and in the next slides, I will explain to you how we build an antibody phage library. So, since we do not use animal cells, we, for our library, use human cells to represent the world's antibody gene repertoire in our library. And we don't need just one person for that, we need many people for that. So we extract B cells from the blood. So normally during a blood donation, this is something that is a waste product. So we can use these PBMCs and extract there the B cell, the genetic B cell information from. So we amplify that and clone that into a so-called phage vector. This phage vector contains the variable heavy and light chain combined and shuffled. So we randomly mix all the VHs we are getting out of the B cells and all the VLs we are getting out of the B cells. At the end, this phage vector is packaged into an M30 phage. This phage carry now the genetic information, the genetic antibody information on its surface and the protein, so the antibody surface, so is on the surface. With this, we achieve a diversity of over 10 billion different antibodies, and this with around 100 donors in our library. So now we have our library. What do we do with that? So short, we go fishing. But as a bait, we don't, we just use our antigen of interest. So the antigen we would like to develop an antibody against. So the whole process takes place in a reaction vessel in our laboratory. There we put the antigen in and our antibody phage library with a diversity of over 10 billion different antibodies. Now, protein-protein action occurs. So we find the antibody on the surface of the phage that bind to our antigen. So here, because all of this takes place in a reaction tube, in an Eppendorf vial, we can also adjust the conditions. So for example, increasing or decreasing temperature, pH, making it more acid or more alkaline. Also, we can increase or decrease salt concentration. So everything you can adjust there, what you cannot adjust, for example, in an animal. So why is that? Could that be of interest? Because in the end, this antibody we are getting out of this phage display approach should work in your assay. For example, your assay is not working in a normal PBS pH 7.4 condition, but in a more acid or alkaline environment, then your antibody should also work under these conditions. So all of these conditions you can influence into phage display approach. So when now antibody and antigen found each other, then we wash all the other antibodies away. That just the one antibody that we would like to bind to the antigen is there. So we use this method to develop a completely animal-free antibody. And for us, it's not enough for just don't use animal for immunization. As I said at the beginning, my vision is also to operate completely animal-free in my lab. So what we also do is we use no blocking reagents that contains animal components. So we don't use BSA or skim milk, for example, in our reactions. We don't use enzymes that are extracted from animals or contain BSA. We have a completely animal-free cell culture. So we don't use FCS or FBS in our cell culture. And we also don't use stabilizers that contains BSA. In the end, I will also tell a bit more about animal-free components. So this will come in some slides. So since this technology gives us the genetic blueprint of the antibody, we can use this genetic information to design our antibody according to our needs. So in this way, we can make a human antibody, a goat, a mouse, a rabbit antibody, IgG antibody out of this genetic information, depending on the needs of your assay. But these antibodies never saw a mouse, a goat, or a rabbit. It's just genetically and antibody engineering. Alternatively, we can also modify the FC parts of our antibodies. For example, we can fuse tags on that or make a so-called silent FC part. What we can also do, we can screen for certain properties of, for example, of the affinity of our antibodies. For example, we can screen for a good K-on value. That means that the antibody binds quite quickly to the antigen, or for a good K-off value, that means the antibody remains attached to the antigen very long. These changes can be made to the antibody at any time, just because we have the genetic information and can then modify the genetic information also afterwards. So when we have already generated the antibody. So now I talked already for around 50 minutes. I would make a 5-minute break now. And afterwards, I would go on with examples of the phage display approach and also with how do I find my animal-free antibody and how do I find animal-free reagents. But now I would make a 5-minute break that everyone can grab some coffee, water, or just go to the restroom. And we will meet us again at 5 past 5 for us, so for German time. So I think it's 11 at Washington, D.C. Oh,
Yes.
okay, perfect. So 5-minute break. So on my timer, it's 5 hours. So I would go on with the presentation. Hope you're all are back from the break. So then we can directly jump in what you can do with phage display. And I choose examples that are hard to get from animal immunization. And these are examples from our lab. So what we did was, for example, we made panning, and panning is the phage display, is the approach to create animal-free antibodies with the phage display libraries, antibody gene libraries. So what we made were antibodies against small molecules. So what we did, we would like to have a specific antibody to that specific structure. So just a ring structure. And there is another molecule that has a similar but not identical structure. You will see in a second which component I'm talking about. So what we can do with phage display is we present the epitope, so this specific ring structure, to our libraries, and we compete with the other structure that we would not like to have an antibody against. So all antibodies in our library that are binding to the one structure will stick to that structure. Antibodies that bind to the other structure will bind to that structure, but will be washed away. So the components I'm talking about are cannabidiol, CBD, and tetracannabidiol, THC. These are really small molecules, so size of 0.3 kilodalton molecules. And with the phage display approach, we developed completely animal-free antibodies that are, so one antibody that binds just CBD, but not THC, and another antibody that binds THC, but not CBD. So with the technique of phage display, we developed antibodies against two antigenes that are really similar and just differs in a small ring structure. This is possible with the approach of phage display. What we also did was and this is what we are at the moment doing a lot, we develop antibodies against antibodies. So what you know mainly as secondary antibodies. So what we do here is we use an antibody as antigen because we would like to get antibodies against the antibody. As an example, we used for our proof of concept study human IgG specific antibodies. So we used human IgG as antigen, and we don't want that it binds to mouse, rabbit, goat, or whatever else antibody. So we compete with these antibodies that the unwanted structures are deselected already in the panning process. So they are also washed away. With that approach, we developed an anti-human Fc-specific antibody that is completely animal-free and detects just human IgG, but not, and this is also what we deselected against, no human IgM, IgA, or IgE, but also it's really specific to human. It don't reacts with other species IgGs, so not mouse, bovine, rabbit, goat, horse, rat, IgY. So we tested against a lot of different antibody structures. What you now can do with that antibody, and we tested that because we conjugated that with an Alexa fluorophore, for example, we made an immune histostaining. So what I did was we just seeded the breast cancer cell line. It's a T47D cell line. And in-house, we also have an animal-free developed human anti-MAC1 antibody. And MAC1 is an epitope structure on the breast cancer cell line. So what we did was just incubating our cells with this human anti-MAC1 antibody. So the primary antibody binds to the breast cancer cell line, and detect this with our animal-free anti-human FC, because we have a human anti-MAC antibody that binds to the primary antibody. Of course, our antibody is coupled to a fluorophore, here in the top with Alexa 488 and in the bottom with Alexa 555. We could nicely stain our breast cancer cell line in a completely animal component-free assay, because what we also did, we used animal-free blocking solutions for that. So it's possible to make an immune histostaining completely animal component-free. So to summarize briefly what I told you in the last few minutes about the different types of antibodies. The most antibodies on the market, sadly, are monoclonal, are animal-sourced antibodies, polyclonal, and the monoclonal ones, but they can at least be made recombinant, which make them a more defined product, even if they are still of animal origin, because they are coming from a hybridoma cell line. But there are also non-animal derived antibodies on the market, sadly a very small number up to now, that can be used in diagnostic and research field at the moment, and they are perfectly standardized and can be easily fit into your assays. So just our goal for future should be to increase the non-animal derived antibodies on the market and to decrease the animal derived antibodies in the market. So just because I will always be asked, so I get always the question, if animal-free antibodies are so good, why do not more people use them? So yes, I told you they're sequence defined, they're reproducible because you know the sequence, therefore they are immortal. You don't have to store them in liquid nitrogen. You can store the sequence on a USB stick and produce them again in 10, 20 years, the exact same product again. So this is a big pro for that. You can produce them in animal free cell culture in a scale you can choose. So also in larger reactor scales. The process I told you about, the panning, just going fishing with the antigen, is a really fast process. So you don't need weeks to immunize an animal. You can do that in a week to generate your animal-free antibody. And I also told you the antibodies can be improved afterwards. So if you need another affinity, for example, or need another FC part to it, that it fits better to your assay, this can easily done afterwards because you know the sequence of your antibody. That makes them a very versatile tool for all for the assays. But, and now the contra comes into place. There are normally higher initial costs for the generation, this I have to say. So you have to pay, so first we have to pay to generate the antibodies, where most of the hybridoma antibodies or polyclonal antibodies are already in the market. There is normally, I said you can improve the affinity, for example, afterwards. And this has to be done if you need a really sensitive assay normally, because the initial affinity of these naive libraries, like I presented it to you, is normally lower than the one of an immunized animal. And we have a new certified we have new products on the market that, depending on the regularities of your institution, normally also needs new certifications. Can be a bit of paperwork using these antibodies. But what makes it a bit, or what makes me sad normally about all of that, is that it feels like a repetition of work, because there are already all the animal-derived antibodies out there, and now doing on the same antigens, animal-free antibodies. So yeah. But this was just a short summary in between. Now let's talk about, again, animal-free antibodies and how you can use them in your assay. So here I would like to show you a picture of the publication of Gray et al. And it's a really nice picture because you see what can be done with antibodies. Or what do you see or not see about antibodies? Let's call it like that, because they call it the antibody versatility iceberg. What you see here is you just see the tip of the iceberg, where you see polyclonal and monoclonal antibodies that you can normally find in your catalog or in a catalog of any supplier you normally buy antibodies from. And there's also this little tip of non-animal derived antibodies. This is what you normally see when you're looking to purchase an antibody. But what is not seen is that there are so many more antibody formats, reagents, fragments of antibodies are out there that you can normally not find or hardly find in catalogs. So the world of antibodies, just because we can generate them and know their sequence, is so huge, and this is normally unseen by most of the research communities. And these are also lost opportunities, because there are all these formats somewhere out there. So just to go back to the formats, I told you that we have the genetic blueprint of the antibodies when they are animal-free developed. And this picture you already know. I showed you what we can do with that. So we can clone them that they look like a mouse, goat, rabbit, human IgG, or modify them. So, but this is just the easy thing. So this is what you can also do with the, so which formats are available. And here, Absolute Antibodies made a really nice, they call it periodic table of antibodies. Here you see really the variant of recombinant possible antibody formats. And this is not already all. So this is also just a short outlook what is possible of antibody formats on the market. And what we see here are the formats here in red that are containing an FC part. So these are the easier examples. Then we have here the antibody fragments that don't contain an FC part in blue. And here just fusion proteins. So let's focus on these two fragments. And here what we see then from the left to the right is that this in one is just the normal IgG antibody, like I explained you the structure. But what's also possible here in the middle or the right part is you can combine several variable binding fragments, so paratopes of an antibody, on one molecule of antibody. So make them bispecific or trispecific even. And there is a whole world of antibody formats out there. But just to let you know, and that there are so many possibilities out there, that what you can just do when you know the genetic blueprint of your antibody. So, but now let's come a bit more to the workshop part of the antibody. So how to find an animal-free antibody. And here I would like to give you two websites first. Please just bookmark them, store them for later. So these are the main source where you find animal-free antibodies. There is this really, this is the best way if you find your animal-free antibody in the recombinant antibodies and mimics database that are released by Animal Free Research UK. Because this is really a database, and I will talk about this in a second. And just as a list of antibodies, you find at the website of the PETA International Science Consortium a list of suppliers of animal-free antibodies. So, and just to go quick on that, how you can find your animal-free antibody. So on the recombinant antibodies and mimics database, you have here the chance to type in the keyword against which antigen your antibody should be. And then you get a list of antibodies out. I will show you in a second. They are rated all, and this is the good thing. These antibodies are rated according to their animal use and their developing and production history. So how are they produced and how are they developed? This database went live just a few weeks ago. And I really hope, and this is my wish, that this data set will grow and we will get more and more informations in there, because at the moment, the thing is on the database, we just, and you see that here live, that we just have around 67,000 antibodies in that database. And I say just 67,000 because worldwide in all catalogs we have over 10 million antibodies available all around the world. So this is less than 1% of all antibodies that are at the moment in this database, but this database just went live a few weeks ago, so the number will increase. So, but just to show you quickly what you see when you type something in there, you will here see category A to G rated the antibodies, and category A antibodies are completely animal-free. So they are animal-free developed and animal-free produced. While category G antibodies, for example, have an unknown source. So this is a straightforward approach to just have a first look if you find an animal-free antibody against your antigen that you would like to replace in your lab here. So then, because I see we're running out of time, I will just go quickly over the next slides. So then there is the list of the PETA International Science Consortium. Here, it's every time worth to click through all the links, because everyone has something else special. So we have my company, Apcalis, here, just saying, if you go to our webpage, for example, you can then see our product list here. For us, I can say all our antibodies are animal-free and have an FC part, so you can easily replace them in your assay. But we just have a limited number of antibodies at the moment. I think we are at 35 or 40, around 40 antibodies we have in our catalog. So it's not much. So we are increasing constantly our catalog, but at the moment we don't have so many antibodies in our catalog. So you have to look for other sources. So Abcam is one of the largest provider of antibodies, so supplier of antibodies at all. You have really a lot of antibodies in their catalog, but I have to say, for them, there's no easy way to understand which of their antibodies are animal-free. And I just give you the hint, you can select there, for example, recombinant antibodies. But if you select for recombinant antibodies, you also get the RepMap antibodies, they call them. So they are normally rabbit-derived. So you really have to have a close look if they are just recombinant by converting a hybrid donor antibody into an recombinant antibody, or if they are recombinant by really an animal-free development, for example, with phage display approach. So this is not easy to find out at the Abcam website. Then we have absolute antibodies. I already showed you their periodic table of antibody formats. So what I can say, they have a lot, really many recombinant antibodies available. And I would say all of their antibodies are recombinant, but there's also no easy way to understand which of them are completely animal-free, because the vast majority of their antibodies is also derived from converting a hybridoma. But if you look for antibodies, and here's just a short example, you can then see sometimes in the text, in the description of the antibody, if they are phage display derived or not. But what is the good thing, and it's also coming from this periodic table I showed you, is that if they have the sequence, they also have the genetic blueprint of the antibodies. You can choose the format of the antibody that you would like to use out of a lot of formats. So you can have a rabbit IgG, a mouse IgG, a human IgG. So there's a lot of formats that you can choose from. So you get a recombinant, so animal-free produced antibody, not necessarily developed antibody. So then we have adipogene here. They provide also animal-free antibodies. They have a nice list if you go to the link, a list of animal-free antibodies, but also there they just have a limited number, but there you're sure they are completely animal-free, developed and produced. And then we have Bio-Rad, for example. And the huge advantage of Bio-Rad is they have their own antibody library, so antibody gene library. Like I explained it to you, how we developed our antibody gene library. They have their own, so it's a so-called UCA library. So they have a lot of antibodies derived via phage display, so completely developed animal-free. And here, just going quickly through that as an example, how you can select an animal, so find an animal-free antibody. So if you go to the Bio-Rad antibodies website, you have to look for UCAL library antibodies. So how do we do that? So for example, I'm looking for a Western blot antibody, primary antibody, and for Western blot, I normally need a housekeeping gene, housekeeping protein antibody. So I just select an actin beta antibody. Clicking on that, getting a list from Bio-Rat. So here's already the first hint that antibodies that have an HCA in the beginning are derived from UCAR library, and there you can be pretty sure that they are developed animal-free. So if we click on that, you see, okay, it's a UCAR antibody, and you see in the description selected from UCAR phage display library. So yay! We know we have our animal-free developed antibody found. Perfect. So already have your smartphone in hands, because next question is coming soon. Because now the thing is, we have found our primary antibody, yes, but just with the primary antibody, we can do nothing. So how do we detect our primary antibody? And what is the best secondary antibody? And now I would like to ask you, and therefore we have a poll. Wait, I have to activate. If you can say, with the information I gave to you, what is the best secondary antibody to detect the primary antibody? Just vote and give ideas what you think is the best secondary antibody to detect our primary actin beta antibody in a Western blot. So I just give you another 20 seconds and I will explain afterwards the results with you. Oh, most of you said an anti-human FAP antibody. So, and some of you an anti-human IgG antibody, some of you an anti-mouse antibody, and have too little information to answer these questions. This question is also given as an answer. So let's have a closer look to that antibody. So what most of you already figured out, and I know I just gave you a bit more than a minute of time, so really good that you already figured it out, that our isotype that is given from the UCAR library is a FAP antibody. So like I explained right in the beginning, FAP antibody is just the arm of the Y-shaped molecule. So, and the other information that is given in the description is that our FAP antibody has a MUC tag and a strep tag at the C-terminus of the heavy chain. So with these two informations, we already know something about our molecule. But now, how do we detect our molecule? So we know here also from the information that we have a human anti-human actin beta antibody. So what we need as a secondary antibody is an anti-human FAP antibody. But what we can also use, because the information is given that the antibody has a MUC tag and a strep tag in the C-terminus, is that we can use an anti-MUC or anti-strep tag antibody as secondary antibody. Bio-Red also give always the link of available secondary antibodies. So if you go to their website and you're not sure which secondary antibody to choose, then click the links and they will provide you with information which secondary antibodies are suitable for this primary antibody. With that information, what is not working are anti-rabbit antibodies, anti-mouse antibodies, and anti-human FC-specific antibodies, because our molecule is a Fab molecule and don't contain the FC part of the antibody. Also, any Fab secondary antibody can be a problem, because what is also given in the text as information is that our Fab antibody has a lambda light chain. So if the Fab antibody, the anti-Fab antibody, is against lambda and kappa light chains, no problem, you can use that. If you have, for example, a specific kappa light chain anti-Fab antibody, this will not work for our primary antibody. So what I just would like to highlight with that is the antibody format is important for your application. So have a look if you find an animal-free antibody, and there are just a few out there. Always have a close look of what is the format of my antibody and how has the secondary antibody, how has it look like, that I can detect my primary antibody, because most antibody formats for recombinant or animal-free antibodies are also just fragments. They don't have to be full. Don't have to be full IgG antibodies where you can use the FC part as detection part. They sometimes are just Fab fragments or scFv fragments with a tag, and then you have to see that you use an anti-tag antibody as secondary antibody. So we are close to the time to the end. This is why I will just shortly, and I know that is really quick what I'm now doing, but you will get my slides afterwards, send it, so you can read that then in detail afterwards also. Because what I totally haven't talked about are all the other reagents in the lab that are also contains animal material and that you can replace by animal free material. And I just need 5 more minutes to go quickly over my slides, and then we have some final questions and a final quiz, and then again a question session. So just quick. So this is why I also skip my question, where what things are normally animal free. But I would just highlight my favorite cookie is an Oreo cookie, and they are vegan. So if you have vegan dairy, Oreos are the best sweet way to go with. But nearly all other components that you use normally in lab have hidden animal-derived reagents inside. So we already talked about antibodies. They are coming normally from animal immunization. But normally, if you work in a biochemistry, biotechnological lab, you also use a lot of enzymes. Or if you work in cell culture, for example, trypsin is derived from porcine pancreas normally. But there is an animal-free alternative that is animal origin-free out there. Restriction enzymes, they normally contain BSA as stabilizers, but there are also companies out there that are use recombinant albumin and are completely BSA and animal origin free. Most blocking reagents in lab are still animal derived. Either it's serum from an animal, it's skim milk, it's BSA or other protein, animal derived proteins. Also there are animal-free blocking buffers out there that are already premixed that you can buy, but you can also try several other solutions that are normally polymer-based, so polyethylene glycols, polyvinyl alcohols, polyvinyl pyridols. Bean 20 already is a good blocking reagent in a certain concentration, but you can also try plant-based protein powders. And here I just recommend just look at the protein powders that are normally used for bodybuilders. You just have to look at the ingredients, just have a look that they have low fat and low sugar concentrations, but these plant-based protein powders can also be easily used in research labs as blocking reagents. Also fetal bovine serum, so FBS, or fetal calf serum, FCS. Okay, it's coming from a bovine, a fetal bovine, so it's for sure animal derived, but there are chemically defined mediums out there that you can use, and there is a publication of Weber et al. They published a universal animal product free medium. So here are really the ingredients named with concentrations you need to make your own animal free medium, and you can try it with your cell line. Already several cell lines are tested with that medium, but you can try that out if that's also working for your medium. And if you're using bacteria in your lab, tryptone, peptones, they are all normally also from animal uh, animal origins. But also there are animal-free plant-based peptones available that you can buy. They are normally from soy. So these were just and really quick examples. There are many, many more animal-derived reagents in the lab. But these were just now the obvious reagents where animal-free replacements are available. So just short take-home message. Animal-free antibody technology aligns perfectly with the principles of 3R. So they are sustainable, and you can easily replace them normally in your lab with the animal-derived reagents you're using. They are defined, the genetic sequence is there, so you can also have a look if you're normally using a mouse secondary that you can get the antibody with a mouse FC if you're using rabbit system. So this is really the benefit of animal-free antibodies, that you can nearly do everything with them and choose the format according to your needs. But I also showed you have to choose carefully your antibodies. So you have to look what they are looking like and what you need as a secondary to detect them. And just last but not least, I would just like to encourage you, if you have the chance and find an animal-free antibody, just give it a try. Just test it, see if it's working in your system. And therefore, I would also go again to the PETA International Science Consortium website. I would like to share that link with you. They have the so-called recombinant antibody challenge. So here you can refund your work if you're trying out in your essay an animal-derived antibody with an animal-free antibody, so you can get refund of that. So if your PA says, no, there's no money for animal-free antibodies, with that challenge, you can get a refund of the antibody to save a bit of money. So now we are really almost at the end, and again, some questions are coming, and I just share for all of you the WooClap event. If you closed your the window on your smartphone, tablet, or browser. And there are two questions coming up now, and one you will just recognize from the beginning. So I just would like to know if how would you rate your knowledge of antibodies now after the webinar? Could I increase the knowledge of antibody you have? And give you 10 other seconds. Then I'll go to the next question. Okay. And I like to see that you have now a more expanded knowledge of antibodies at all. But now I would also like to know, after this workshop, do you consider using animal-free antibodies or reagents in your work? I'll give you another 10 seconds. And I really love to see that you're open to the idea of using animal-free antibodies and reagents in your work. So I will close that session now. And now we are really finally nearly at the end. So there is a little quiz just to check shortly with 4 questions. Yes, your knowledge about animal-free or antibodies at all, let's say. So at each question, there's just one correct answer, and you have 90 seconds per question to answer. And then afterwards, we are done with that part. And then there is space for answering questions. So, but first start with the quiz. Okay, because number is of participants is now we have some increasing, but I think I will not go over the whole time. So 10 seconds, and then I will go for the next question. So just show. So 82% of you were correct with the answer. So let's come to the next question. So give you another 10 seconds. And 85% of you answered this question correctly. So the third question. We'll give you another 10 seconds. Okay. And 91%. Wow. Perfect. And last but not least, the final question of this workshop. So give you another 10 seconds. Yeah, also 91%. So thank you so much for your attention and the participation in the WOOC Lab polls. if you would like to stay in contact with me or Carlos, just follow me or the company on LinkedIn. And if you have any questions, feel free to put them in the QA chat. I hope we still have a few minutes to answer them, and I'm so sorry that I'm over the time. I know I should end it, yeah, a bit, yeah. I'm sorry for being over the time.
No
So,
worries, Esther. It was wonderful. And there are several questions, but we can go through them quickly. It was very, very interesting. And yeah, I saw from the questions that people were very engaged. The first, maybe I can just say they can email you. You just mentioned that because there was one question about getting work experience, maybe also at your company. I don't know about that. If you have
yep.
capacity, they should just reach out, right?
Yeah. Just email me, follow me on LinkedIn, text me on LinkedIn. Yeah, so LinkedIn is the best social medium for me because I'm not at Facebook, Twitter, or X or whatever. So LinkedIn is my social medium. But there you can text me if you would like to. Yes, for sure.
Well, thank you. So, okay, there are many questions. Some of them I tried to answer already because I don't think we get through all of them. But they were easy. I mean, not that. I hope at least. So the first question, can we create artificial antibodies to create a non-lethal rabies paper strip test?
The thing is, what is meant by artificial antibody? So antibody.
I think what they mean probably is animal free. I think that's
Yeah.
probably what I was.
If artificial is animal free, then yes, for sure. So just need so to create an animal free antibody, the only thing you need is the antigen available. And for rabies, the antigen is available because I guess for rabies it's also the vaccine on the market. So it has to be there in a pure format. So yes, there should be no, yes, it should be totally possible to create animal-free antibodies against rabies.
Okay. Then another question, you explained it a little bit, but just to clarify again, they were asking if the animals are killed after each round of antibody generation, or are they used several times? And maybe you can also speak about what that means for the animal.
Yeah. So I know that just from literature, I have to say, I have no working experiments because since my studies, I refused any animal experiments. So I never did animal experiments and I would like to stay with that. So all I know is from literature, so not by personal use. But what I know for the development of polyclonal antibodies, an animal is used several times. So you inject several times an antigen and get the serum out of them. They can recover in the end, but then they get used for another they are used for another injections. They get again drawn the blood. Yes. For hybridoma development, normally the spleen is used from the mouse. And if you take the spleen, normally the mouse is killed. Yeah. So
Yeah, thanks a lot. There was also a question that I already thought wasn't relevant because you were asked when you're using the animal-free antibodies. Obviously, you're only using animal-free.
yeah.
So yeah.
Yep.
For how long can one use the hybridoma cell line? Is the question, how long can you use that cell line? I guess with time, they can be subject to spontaneous mutation that can affect the antibody's quality. That's the question of the next.
Yes, that is true. So a hybridoma cell line you normally store in liquid nitrogen. So also there, spontaneous mutations can occur when you recultivate them. They can then also, if you in the first glance don't think you have a monoclonal antibody, also what I showed you with that an additional light chain can be produced so that you don't have any more monospecific antibody. All this can occur to a hybridoma cell line. This is why, if you would like to start the conversion to animal-free antibodies, best is to first convert your hybridomas. And it's easy if you have the hybridoma cell line. It's just an RNA extraction to get the genetic information out. And then you have the genetic information. You can store that on a USB stick on your cloud servers or wherever, and you have the genetic information that can you can have forever and produce the antibody always in the same format again. So yes.
Okay, and then it was the question about in silico approaches. Obviously, you utilize those. Can you speak a little bit on that? How they can be utilized to create new antibodies or optimize antibodies?
Yeah, so also in silico approaches, and I guess within silico approaches, you mean the new AI tools coming up that really calculate antigen, so epitope, paratope binding. So this is coming up more and more. We are just at the beginning to figure out what is all possible with that, but I think this is the future also, so that there will be more programs on that, and that I'm really looking forward for the future and what's coming up within silico approaches there.
Yeah, it sounds very amazing. I think that I don't know if we have time, Catherine. You have to say if we have time to answer the last questions, or you rather want to just briefly talk about the next workshop we have planned. Michaela, maybe I hand it over to you.
Yes. Hi, we are at the end of time. We're at the top of the hour, but I just wanted to thank everyone so much for joining us today. And thank you, Esther, for giving such an engaging presentation. I want to take the time to announce our next workshop in the series. It's on exploring brain MPS, organoid intelligence, and career pathways. It'll be in two parts. The first will feature a presentation with Dr. Itzi Morales-Pantoja, and the second will be a career panel that invites representatives from academia, industry, policy, and government in, where you will have time to ask questions of people working in different spaces in animal-free research. Thank you so much again for joining us. I think if we want to answer the rest of the questions for people who want to stay on a little later, and if Esther is willing to, we can probably do that. But we are at the top of the hour for people who are ready to go. Thank you so much.
Awesome. So Esther, if you still have, I don't think it's too much left, but if you still have time, we could go through the rest.
Yeah, for sure.
Because I think, yeah, we are always happy about receiving questions. You know, for some webinars, people are very shy and then they don't ask, and yeah. So so can you say something about the market cost of such animal and non-animal antibodies? Obviously, that's a good question, especially for early career scientists that sometimes have to persuade their PIs to even make the change.
Yeah. So what I can say, and what you can also figure out when you go to the to your normal suppliers of primary antibodies, normally the cost of primary antibodies is nearly the same when you're using animal-derived and animal-free antibodies or recombinant and animal-free antibodies. I know there are companies out there where you get pretty cheap animal-derived antibodies, primary antibodies. I know that. But then you have to ask yourself, how was the animal treated to get such a cheap antibody? So normally the price for primaries is nearly the same. For secondaries, I have to say, the price of the animal-free antibodies we're at the moment developing are more in the range of a primary antibody than a secondary antibody. But again, secondary antibodies is serum drawn from the blood of animals. So it always, the cost of that yeah, it always depends where the animal had its life. Let's phrase it like that. In Europe, we don't have, as far as I know, we don't have much animal farms anymore because for them it's too cost intensive. So most of the polyclonal antibodies we are getting are from Asian market, as far as I know. So there the costs are a bit cheaper, yes, but you don't know how the life of the animal is there. So yeah, let's say primaries, there are equal costs. For secondaries. We have slightly increasing costs, but I would emphasize there again on the antibody challenge from the PETA Science Consortium. So if you would like to test it, you can get a refund of your animal-free antibodies if you apply for that challenge. And this is probably also a step to go if you would like to have something in hands for your PI to yes.
Yeah, definitely. So it's basically the cost for the animal versus the cost, for us. How ethical do we want to be in our research? Yeah.
Yeah.
Another question, the attendee has heard that the industry does not order animal-free antibodies via phage display because nobody should know on what they are researching. Is that correct?
I never heard of that, and I have to say, so we also have, so we are also contract service manufacturer. So we also have our services from industry, and they're just confidential clauses. So I cannot talk about all what we are doing research on, but I can say that also industry is getting more and more into animal-free antibodies also. Yeah.
That's good news. Then there's a question, another question, is it possible to multiplex with non-animal antibodies? Because based on the explanation you gave, it seems they will all require anti-human secondaries.
No, I just showed the example with the anti-human secondary. So there will be coming soon also anti-mouse, anti-rabbit, whatever animal antibodies. Multiplexes will be then possible if so we call our anti-human I showed you a multiclonal. So there are secondary antibodies mimicking a polyclonal serum just by mixing defined monoclonal antibodies together. And with this kind of approach, and this is why it takes so long to bring that into market, because it needs an extensive characterization of the secondary antibodies. But with this approach, you can also multiplex signals, and there you can then replace your animal-derived antibody with an animal-free secondary antibody. And there's just the anti-human at the market, but more coming soon.
How plausible would expanding donor B cells in culture be and induce somatic hypermutation to improve affinity before library generation?
This is a really interesting question. I'm not so much into the so I have no experience in culturing B cells, and but it seems like it's mimicking the immune system when challenging it with several immunizations, for example, with the same antigen. So just inducing that in cell culture is a really, really interesting topic. Never thought about that, but this is, I think that could work out, yes.
Yeah, and this person actually wanted to reach out to you anyway, so you can discuss further. Yeah.
Yeah.
Okay, then more questions to come. There's a question about regarding the process. How do you mitigate the overrepresentation of certain antibodies sequences in the display library, which can bias the selection process? And how do you ensure the identification of rare but better candidates?
Mm. We while let's phrase it the other way around. During the panning process, we can work really strict on the anti-gene binding by adjusting, for example, the pH, the salt concentration, temperature, so the whole environment conditions. And these are normally so strict that we get that we can wash away pretty easy antibodies that are overrepresented, but not epitope binding relevant from our phage display approach. So with all of our conditions during the panning process, we are already pretty strict to get out the best candidates. But also there are sometimes antigenes where we get several binders out there, and just to be sure to not overrepresent, to just choose the overrepresented ones, but probably also the rare one that is but then the perfect candidate. There are also in the newer technologies like NGS sequencing, for example, makes our life more easy, because there we see what we have in and can also look at several thousand sequences instead of just 1, 2, 3 sequences. Yes.
And the second question by this attendee is there a different regulation for the usage of animal-free antibodies in clinical trials?
Let's phrase it other way around. In my talk and workshop, just talked about diagnostic and research applications, because this is the field where we have under 1% of all antibodies available are animal-free. The total opposite we have in the therapeutic field, because nearly all therapeutic antibodies on the market are already animal-free. Because if you have a therapeutic and you would like to inject it into a human, it has to be standardized, it has to be controlled, it has to be so that there are already all the security steps, and it has to be ensured that the sequence is known, that you can produce it all at the same time. So all what I aim for to use it in research is already done for therapeutics. So they are already an established protein on the market, the animal-free antibodies, but not in research and diagnostic fields.
Yeah, that's kind of sad, actually, right?
Yeah.
Okay, and then there was one more question about the different how are your antibodies different from the SUMAP recombinant antibodies offered by Merck? I mean,
this is a good question, and I think I'm not so much aware of the SUMAP antibodies that I can answer this question. Sorry.
at least they're both xeno-free. I guess that's an
Yeah.
important message we want to give in this
Yep.
in workshop, right?
Yeah.
Okay, and then I don't know if you can answer this, but about the US, they were asking because of the tariffs now. Do you know where to get animal-free antibodies made in the USA? Companies, maybe one or two. Don't worry if not,
Yeah.
we can find it online, right?
Yeah. I'm not so sure right now. So I don't know if EpCam also has the manufacturer in US, for example. They're normally UK based. Here, I'm not sure at the moment.
Yeah, I saw
Probably, yeah.
ADMAP is one. I don't know how big that is in Antibodies Incorporated. I mean, but I'm sure if you do a proper search, you can definitely find that.
I would just recommend to the attendee, look at the list. Also, again, PETA Science Consortium. So their website, they have a list of a lot of manufacturers. I just went through the first 5 or 6, I guess. But there's a long list, so probably click through all of them if there are also an US-based company. This is this is my suggestion to this attendee.
Thanks a lot. So there was a wide range of participants and also the knowledge they already have. I saw, and there was a question about an entry level, book you can maybe recommend. I don't know if that's.
Wait. I just go to my bookshelf, and I know that's just wait, I have to. Where do I get my
Okay. But
shader off?
maybe we can put it in the follow-up email. Michaela
Yes.
will send around your
Yes,
slides.
and I have also several other books. So there are some books about especially recombinant antibodies that yes, I would make, and this is a good point. You get my slides anyhow in the end, and I will also make a slide of literature where you can look this up in literature. Yes.
Okay.
I'll do that.
Yeah, wonderful. That's great. Michaela will send everything. Michaela and Catherine, you want to come back? Yeah.
Catherine had to go to another meeting, but thank you so much for staying on to answer the remainder of the questions. I think a lot of our participants really appreciate that. Many of them are still here and did not leave at the top of the hour. So thank you again, Esther, for the great presentation, and thank you, Kat, for helping organize this event. We look forward to seeing everybody at the next workshop in July.
Take care, everybody. Thanks a lot.
Bye.
Thank you.